Hey folks, welcome to Chapter 9 of our course, where we're going to focus on neurodevelopment. Now, if you're a psychology major, you might be aware that you could take multiple classes on developmental psychology and how we develop our cognitive functions and our psychological functions of various kinds. Here we're going to be focusing on how your nervous system develops. That'll have implications for psychology, but we're going to hit some of the high points of particularly neurodevelopment and not necessarily all of developmental psychology. So across a few videos in this chapter, we're first going to do early basic neurodevelopment in 5 phases. We'll cover that in this video. Then we're going to talk about what happens after you're born, so postnatal cerebral development. How does the brain continue to grow? How does experience affect brain development throughout early childhood and adolescence? Um, then neurogenesis, whether we develop new neurons as adults or not. And then we'll talk about just a couple of disorders of development as examples that your book highlights. So let's get started on the first part, our 5 phases of neurodevelopment. And here they are. You can go ahead and pause the video if you want to take notes on these 5. Students in my class should be aware of the 5 in the order in which they follow. And we're going to walk through these individually as we go, but you should be aware of the 5 and the order. So let's get started. At the very beginning of neural development, we're going to start with embryos, right? So there's embryonic cells that we're going to be getting from sperm and egg coming together. Those embryonic cells are stem cells. And they're what we call totipotent, which is like total and potent, all powerful, right? They're totipotent, which means they can form into any type of cell. So your embryonic cells start to divide, and they start to divide in their stem cells. They can then become muscle cells or skin cells or neurons, right? They start to differentiate during development so that they are no longer totipotent stem cells and they start to specialize. We begin to form a neural plate on the dorsal surface of the embryo where we now get multipotent stem cells that can form into any neural cell. Okay? So this is specialization is going to happen for other parts of the body as well, where we're no longer where we're forming muscle cells to form muscles, right? Skin cells to form skin. And so we're going to start to specialize as we go. The neural plate is what we refer to as a specialized area that begins to form just neural cells. During early development, within the first 24 days of development, these start to specialize at the neural plate, then begin to form a neural groove, and then eventually form what's known as a neural tube that eventually is going to be your spinal cord, and then eventually your brain is going to develop off of this. Okay, so within the first 24 days, we're specializing neural cells into this tube that's eventually going to then build a brain off of it. Okay, so it's very early stages of neural development. Your book reiterates if you're not quite sure what a stem cell is. So your embryo cells are stem cells. They can become any number of different cell types, blood cells, neural cells. I think this is a fat cell. I don't know. But they can also replicate themselves, right? So your embryo cells can undergo mitosis and create more. Okay. Once they specialize into multipotent neural cells, they can still replicate themselves and make more neural cells. That's how we get to the second stage. Okay, so we started to specialize a neural plate and a neural tube, and then once we have neural stem cells along the neural tube, they can start making more neural cells. Okay, and this happens very fast. This is what's known as proliferation, right? So if I'm proliferate, right, then that means I'm making a lot, right? So we get a rapid increase in neurons from those stem cells in the neural tube. We start from 1 of course, to over 100 billion by the time a baby is born. That's about 15 million neural cells per hour or 250,000 per minute. So this is rapid generation of new neural cells. Once we get all those neural cells, they need to go somewhere, right? So they don't just stay where they are. They'll actually move along the neural tube, and then they'll start to aggregate together to form a brain, right? And other parts of your nervous system. So we create new neural cells, and then they undergo migration. Okay, that migration is going to be radial, sort of to the outside of the neural tube to build it up, and they'll also move down the neural tube to gather into a certain location. This can happen in one of two different ways. Number one is that the actual nerve cell can extend a process and move, and it gets directed toward chemicals or away from chemicals to find its way. Okay, so down the neural tube, it's actually going to move the cell itself, following chemicals, which is kind of cool. That's called somal translocation. So the actual neural cell moves. It can also be moved or migrated along glia. So there's another nice role for our glia that are not neurons themselves, right? But they're going to move along glia to form, to do radial migration. Here's an image from the textbook that we've been using on somal translocation. You can see the cell extending a process and moving itself along. Okay, so they're going to do this radially and also down the neural tube tangentially. Okay, so this is how your neurons are going to get from where they're created to eventually moving somewhere where they can build up parts of your brain, for example. Glia will form structures like this so that the neurons can actually move along them, which is cool. So the neurons migrate, and the reason they migrate is so that they can aggregate or come together, right, to form neural structures. So if I want a big frontal lobe, by the time I'm born, I'm going to need a bunch of the neurons to migrate and then aggregate together in a location, right? So they're going to build up the neural tube in a particular location that's going to then form eventually different parts of my brain. Okay? So they migrate so they can aggregate. There are molecules we've identified called neural cell adhesion molecules that actually hold the neurons together. So they migrate somewhere, and then the molecules that get developed actually pull them together so that they'll adhere and then build up together and aggregate. We also have gap junctions as the neurons come together. They're actually communicating through gap junctions. So they don't have synapses yet. They don't have neurotransmitters that are crossing, but they are communicating directly via gap junctions. Like I just said, once we come together, we migrate and we aggregate. We don't have synapses yet. That's the next step, right? So we start to build up a hindbrain, a midbrain, a forebrain, and then we're going to start to draw connections once we've aggregated together. Draw connections close by and far away. So the first thing we need to do is grow those axons. So we've got the cells that have come together. We're going to grow axons off of them so that we can connect to other neurons. You got these things called growth cones and filopodia. These are filopodia, almost look like webbed feet, right? They actually come off of the neurons themselves. These are going to reach out and form axons. There's a bunch of research on how these axons form and how they get where they're going. So, big question arose given that we don't have axons and then we grow them and then one neuron will connect to another one. Are they going to a specified location or do they just grow haphazardly and we learn to use them? Okay, so research on this was done with frogs, where they matched the neurons in the retinal ganglion cells, followed them along the optic nerve to the midbrain, and they could map the pathway from the retina to the midbrain, right? And the cool thing about frogs is that if you cut their optic nerves, they'll grow back. So what Roger Sperry did was he took the frog's eye and he flipped it upside down. Okay? And if you flip the frog's eye, and you don't cut the optic nerves, right? What you'll notice is that they're going to be mismatched, right? So this part of the retina is now going to be going up here, where it should be this part of the retina, right? And the frog um, can't catch flies anymore. It can't identify where things are in space, right? So I flipped the eye upside down. That's a problem. Sperry then cut the optic nerve, right? And then rotated. And what he found was that the optic nerves would grow back to where they're supposed to go. Okay, so they grew back, not just straight back, they grew back to their targets. Okay, so specific chemicals seem to attract axons to specific locations. So when we cut the optic nerve here, the optic nerve didn't just grow back straight back and hope for the best. It grew back along chemical pathways, right? And so the rotated eye was still rotated, and the pathways went to the same place, and the frog still couldn't catch its target. Okay, so this suggests that the axons that are grown, once our neurons find their place, are going to targeted locations. They're almost predetermined to some extent, right? So your optic nerves are going to go to where they're going to go based on chemical properties. They don't just randomly go back and hope for the best. Once the axon actually gets there, so it follows along those chemical pathways to find its way to the target, we then form synapses on the end. This is called synaptogenesis. Genesis is a beginning, right? Beginning of synapses. Astrocytes, our good friends astrocytes, these star-shaped glia, actually help with synaptogenesis. So one neuron's connecting to another one, and the astrocytes can reach out and draw connections between them to help communicate while the synapses are getting started and help them to survive. And we get chemical cues, right? So those chemical cues that drew an axon to its target, similar chemical cues are going to allow that synapse to continue to keep one axon close to a dendrite on the other side, as opposed to being drawn away. The final stage of neural development, so we spent all this time building a neural tube, right? Induction of the neural plate. We proliferated billions of neurons. They migrated and aggregated together to form brain structures. We then built out axons and synapses so that we could connect those neurons. And then the final stage is that about half of them die. Okay, so we spent all this time and energy building a bunch of neurons, proliferating, migrating, aggregating, forming synapses and axons. And now half of those neurons are going to die. So why do half of them die? Well, it turns out that a bunch of those synapses, even though they're drawn through chemical connections, a bunch of them are going to be imperfect. We're going to make too many. Okay, we're going to make too many neurons. We're going to have an extra set, and we're going to have too many connections. And we have to prune those to be more efficient. One question is whether they die actively or passively. It turns out that neurons passively dying leads to inflammation, and inflammation in your brain is quite bad. We don't have a nervous system, we don't have an immune system in our nervous system to deal with inflammation, and so that can cause brain damage. So much more common is that the neurons are actually going to be programmed to die. So if they don't get the right chemical signals from a well-formed synapse, they'll undergo apoptosis, which is programmed cell death. So the cell will actually die, and then it will be broken down into parts that will get recycled, as opposed to just dying passively. So your neurons are kind of genetically pre-programmed to die at a certain time if they don't get any certain triggers. If there aren't survival chemicals like neurotrophins, then your neurons will die. It turns out that's a good thing, right? So having too many neurons, having too many synapses is inefficient. It's not good for communication. And so having a programmed system of neural death helps us to have good neural communication. After we form those synapses and the neurons die, we then rearrange our synapses. Okay, so from a kind of chaotic, diffuse pattern of synapse connections, we get a more targeted, more focused pattern of connections. Okay, we want to focus the output to have fewer synaptic connections, to have a more systematic, more reasonable communication system between neurons. So that's our first early stages of neural development. Basically, all of this is going to be happening prenatally before you're born, and a little bit afterwards with some of the neural death and cell rearrangement is going to continue, but most of this is going to happen before we're born. In the next videos, we'll talk about what happens after you're born, through experience, and later in life.